Good days everyone! This video is made as part of the final project for Bioprocessing Technology and Downstream Processing course. In this video, we will be talking about upstream and downstream processing of algal oil rich in DHA and EPA. Let's start with a little background information. Omega-3 fatty acids, specifically DHA and EPA, are known to play numerous roles in maintaining human health. Here, we choose microalgae due to its many advantages.
Moving on to species and parameters. For this project, we chose heterotrophic microalgae due to its ability to efficiently produce metabolic energy. The most important property of this species is temperature, which is adjusted for growth or fatty acid accumulation depending on the needs. These are three strains that commonly used for DHA and EPA production due to its capability to produce high yield of fatty acid, with each species having different growth parameters such as nitrogen limiting medium, salinity level, and certain DO level for their growth.
After knowing the different properties microalgae species have, now we'll get to know the bioreactor used to grow the algae. Testing phototrophic and heterotrophic algae requires different reactor. Photobioreactor produce algal oil in high purity, but due to self-shading, it requires costly maintenance. In contrast, fermenter is more cost-effective, but it's prone to contamination, requiring additional steps to ensure product purity.
Next is for the reactor control system. Several condition parameters need to be controlled and monitored, such as the temperature, pH, dissolved oxygen, and the biomass weight. For heterotrophic microalgae, they are grown using the conventional fermenter. A combination of feed-forward and feedback control system is suggested due to the proneness of microbial contaminations. As for the heterotrophic microalgae that utilize the PBR, only need a feedback control system due to the low chance of contamination.
Moving on to biomass separation. Two common methods that are used are dissolved air flocculation and centrifugation. In dissolved air flocculation, fatty biomass are separated using the principle of hydrophobicity alongside with the air bubble produced by agitation. Centrifugation produces higher fatty acid recovery by 68% in comparison to flocculation with only 52%. However, this method requires a combination with other techniques to increase the separation efficiency while reducing costs.
Moving on to biomass optimization, this process can be done by removing water content to extend the biomass shelf life, and commonly used method is spray drying. This method is considered most appropriate for human consumption because the product's quality can be maintained, but it also requires a cost for it. Another one is the cyclonic separator. It is known to be highly efficient for drying process, but is also prone to cell disruption due to high pressure automation process, thus will degrades the product's quality.
For cell disruption, according to a study, the microwave oven method is suggested due to the 2 times higher yield compared to the other methods. Using the microwave oven method, the cell disruption was easy, effective, simple, and penetrate the cell wall better than the other methods. There are also limitations to the microwave oven method, as it releases not needed cellular components, lack of selectivity, and therefore additional purification step is necessary.
Moving on to purification and refinement. In purification, urea complexation is the method of preference as it is simple, low cost, and have high efficiency. The process starts with converting 3-acetylglycerol into fatty acids. The free fatty acids are then mixed with an ethanolic solution of urea, which promotes an efficient separation of free fatty acid fractionation. Next, membrane filtration is done to acquire the final product. Moving on to refinement process, the refinement process functions to remove phospholipid, unsulfidied materials, particle materials, and chemical contaminants. The process goes from deguming, caustic refining, which then require washing, physical separation of aqueous phase, and vacuum dryer. Next is bleaching, which then followed by winterization, and lastly, deodorization, which is then followed with cooling down.
With the whole process covered, now we'll take a brief viewing on the outlook of algal oil production. Following the emerging trend of convenient consumption and better nutrient delivery, algal oil could be formed in the nanoemulsion or capsule. This enables DHA and EPA to be better absorbed and integrated into various diets. The following are the key takeaways from the algal oil production processes. Due to the high demands of DHA and EPA, heterotrophic algae have the potential to utilize carbon and nitrogen to produce algal oil using a fermenter. In downstream processing, spray drying could be done to the biomass before being centrifuged, which gave better separation compared to dissolved air flocculation. To recover the fatty acids, cell disruption utilizing microwave oven is the most efficient compared to others. This step is continued by urea complexation to further purify the resulting fatty acids. Due to its capability, further researches are recommended to increase the production efficiency as well as replicating the process to other similar compounds. That's the end of our presentation. Thank you everyone.